We'll probably see a question on the first exam from the syllabus, so make sure you know the syllabus. And it's very general, but make sure you read it and know it. And I videotape everything, but I do expect you to come to class. So the videotapes are there to hopefully help you in those rare times when you have to miss class, or to help you get on top of things that you need to get on top of. So it's very good. Anybody going to exotic places over the break? Where'd you go? The Caribbean. You didn't come back with much of a tan. You did, okay. We just had to, I had to get even somehow for those of us who couldn't go to the Caribbean. Anybody else go to the Caribbean? Beaverton. Where's your tan? Yeah, oh well.
Kind of rainy.
Nebraska. It was a little rainy. Nebraska? Are you from Nebraska?
No, the family there.
Oh, so people wouldn't go there unless they were from there, or yeah. No Nebraska jokes here, right? I've got a whole bunch of Nebraska jokes for the term, so you guys will be all set to go tell your friends from Nebraska all about Nebraska. And so you had a fun break?
I did.
Did you kick somebody?
Very hard.
Or they kicked you very hard, it looks like. Okay, so I look forward to a fun term. This is actually the first big class I ever taught. This is, I think, the 16th year I've taught this class, so I've been around a long time doing it. And it's a class that I think most students find is not as difficult as the class last term. There are no calculations, which is why you see no recitations, which is why it's a 3-credit course instead of a 4-credit course. So there's a lot of stuff to know. That doesn't change. This is biochemistry, right? And I still talk just as fast as I did last term, so I'll try to keep that as slow as I can. What we will see in the term is it's kind of a schizo term. Okay? It's a little schizophrenic, even more schizophrenic than I am. And that is that the first half of the term will be almost completely metabolic pathways. You guys got sort of a start in the metabolic pathways. That microphone, can you hear me back there? Is the microphone coming out?
I think it's on.
No, it's supposed to be on, but let's see, it's not. The bad thing this term is I end up fighting the microphone half of the term because they have each classroom has its own stupid microphones. This one, for some reason, is always out of juice. And I'm not quite sure why. This is the first day of class. Not a good sign, right? What's that? That's not my job, as they say. All right. Okay, I guess I'll talk loud today, kind of like you guys are right now. Okay, so I think what you will find this term is that you will really, I think, hopefully see the real connections now between metabolism and things you're doing with your body. Okay? And you started to see those last term with respect to, people are smirking at me like this was a joke or something. There's no joke coming. This is, seriously, if you're running, if you're jogging, if you're exercising, if you're not exercising, if you're eating too much, if you're eating too little, I think all these things will come to make sense for you with respect to metabolism this term. You saw the beginnings of that last term. You saw how glycolysis, gluconeogenesis, and glycogen metabolism all were tied together, and many of those same ties that you saw last term are going to carry over into this term, where you're going to start seeing how energy really is managed in the body, and how fat metabolism affects everything, and how exercise affects everything, how oxygen affects everything. And so I hope that what you walk away with this term is a very good understanding of those processes. I happen to think it's one of the most exciting areas of biochemistry, and it's one that people take for granted. So hopefully we won't have that happen. Gesundheit. All right, so let's go ahead and get started with enough of this silly banter here and talk about the citric acid cycle. So the citric acid cycle is usually taught after people have covered sugar metabolism, which is what we're doing. We changed the course a few years ago. We used to teach citric acid right next to glycolysis, but that meant that people learned that last term, then they had to come back this term and learn glycogen metabolism, and I think as you probably saw, glycogen metabolism and glycolysis and gluconeogenesis are pretty intimately tied together. So we've swapped it now where citric acid cycle is taught this term, and it's a much better arrangement in my opinion. The citric acid cycle is something you probably had in basic biology classes, and one of the things that I will tell you about the citric acid cycle is it is one of the major energy pathways of the body. Glycolysis played a role in that, but mainly because it's setting up the citric acid cycle. If you recall in glycolysis, there were only 2 ATPs and 2 NADHs that were net products of glycolysis. That's not a lot of energy. We're going to see that there's a lot more NADHs and ultimately triphosphates that are produced as a result of action of the citric acid cycle. So that's going to be a big kick. The other pathway, the other products of glycolysis you probably took for granted were the 2 pyruvates. And it's the 2 pyruvates that will become very important for us as we start thinking about how those guys get oxidized. When we think of the citric acid cycle, it's one of the major oxidative pathways of the body. One of the major oxidative pathways of the body, meaning that it generates a lot of NADH and FADH2. Okay? In addition, as a result of the oxidation that happens in the citric acid cycle, every time the cycle turns one time, a carbon dioxide is produced. So when we take those 2 pyruvates from glycolysis, every time it turns, we get 2 carbon dioxides. Every time we turn those 2 times for glycolysis, we get 4 carbon dioxides produced. Okay? So the citric acid cycle is a major source of oxidation, it's a major source of carbon dioxide, and it's a major source of NADH and FADH2. And in addition, you'll see that it also directly produces GTP. So all these things happen as a result of action of the citric acid cycle. The cycle is, of course, and by the way, you'll notice this term I frequently provide notes, at least for about the first 2/3 of the course. Those are for your knowledge and information. I'm not going to go dig things out of them that I haven't talked about in class. So these are just to supplement your knowledge, and if they're helpful to you, great. If they're not helpful to you, don't sweat it. Schematically, this is what the citric acid cycle looks like. The citric acid cycle is one of 2 major cycles in our body that are cyclic. The other being the urea cycle that we won't talk about. Okay? But the citric acid cycle is what I like to think of as a wheel. It starts with a 4-carbon molecule. And by the way, there's no start in a wheel. So traditionally we talk about it starting at a certain point. But the reality, there's no beginning and there's no end. Okay? Traditionally we talk about the start of the citric acid cycle as starting with a 4-carbon molecule called oxaloacetate, which you saw last term was produced during gluconeogenesis. And oxaloacetate becomes joined to an acetyl-CoA, and I'll show you the reactions for this, so just kind of relax for a minute. Gets joined to an acetyl-CoA to create a 6-carbon molecule. And that 6-carbon molecule, through 2 decarboxylations, is broken back down to a 4-carbon molecule. Oxaloacetate is a 4-carbon molecule. And the rest of the cycle is involved in regenerating that oxaloacetate. So we can think of this as the first part of it involving breakdown, that is the decarboxylations, and the rest of the cycle involving regeneration of the oxaloacetate. That's basically what's happening here. You'll notice that there are 8 electrons that are produced. That is, oxidations are occurring. Oxidations occur with electrons in pairs. So that means there's 4 separate oxidations that are occurring in the citric acid cycle. We only saw 1 in all of glycolysis. In addition, each time the cycle turns, we get 1 GTP produced, and we'll see about how that goes as well. Now, I have decided I'm probably not going to make you memorize all of the intermediates in the citric acid cycle. Okay? There you go. In terms of structure, you will need to know the names and the enzymes. You need to know the names and the enzymes of all the intermediates in the citric acid cycle. You need to know where NADH is produced. You need to know where FADH2 is produced. You need to know where GTP is produced. But you will not have to draw structures as a result of my covering things in the citric acid cycle. And by the way, for those of you who haven't had me, I try to do as little in terms of memorization of structures as I can. There are a few places where you will need to memorize structures, and I will tell you what those are. Okay, welcome. All right, so that wasn't a joke, that was a welcome. Okay. Here's a little bit more detail about that cycle, and the cycle looks like this. By the way, speaking of coming in late, I talked to a dental student just a couple of days ago, and she said they had this really cool thing that they did with their class. I'm not going to encourage you guys to do this, but I'll tell you because this is what the dental students do. So whenever somebody comes in late to class, and that doesn't bother me at all, by the way, I have no problem with that. But when somebody comes in late to a dental class, everybody in the class goes, apparently. So might be an interesting tradition to start at OSU, wouldn't it? It'd be fun. But again, it doesn't bother me. So, all right. So this shows some more detail about what happens in the citric acid cycle. There's our sort of starting compound. It's a 4-carbon compound. We add 2 carbons to it from acetyl-Coa. And we'll see that the acetyl-Coa comes from pyruvate in glycolysis. And we'll cover that as one of the first things that we do. There's C6, indicating it's got 6 carbons. We see 2 decarboxylations, each one producing an NADH, one here, one here. We're back down to 4 carbons, and then the rest of that regenerative process involves production of GTP, production of an FADH2, and production, finally, of another NADH. So 4 oxidations with each turn of the cycle. Okay, this is a figure that probably may not make a lot of sense right now, and I'm just going to briefly mention it just so that it gives you an overview of what we're going to be talking about and doing. And this overview shows you that many things ultimately get oxidized in the citric acid cycle. The citric acid cycle is another example of what we describe as a central metabolic pathway. The central metabolic pathway is central to cellular metabolism. It's found in virtually every cell on the face of the earth. It's not absolute, but it's pretty close to it. And it involves oxidation of byproducts of fatty acid metabolism, byproducts of glycolysis, sugar metabolism, as well as byproducts of amino acid metabolism. Well, what do I mean by byproducts? The byproducts are each of these pathways can ultimately produce acetyl-CoA. So acetyl-CoA is a byproduct of each of these pathways, and in the citric acid cycle, that acetyl-CoA is oxidized. Okay? Now, what the rest of this figure shows you is that we don't directly produce ATPs in the citric acid cycle. They're produced as a result of the electron potential that is generated by that oxidative energy, and that potential is realized in the electron transport / oxidative phosphorylation processes that we'll be talking about probably next week. Okay? This also shows you, although it's a little hard to see, that the cycle itself occurs inside of the mitochondrion. Inside of the mitochondrion. Those of you who recall anything from last term at the very end may recall that when I talked about gluconeogenesis, there was one molecule that was not made in the cytoplasm. Which one was it? Oxaloacetate. Aha. So it's very conveniently there inside of the mitochondrion, which is where this overall process is occurring. And we'll say a lot more about that. There's one exception to that, and it's actually an enzyme of the citric acid cycle that is located not dissolved in the liquid of the mitochondrion, but rather in the membrane. So one of the enzymes is embedded in the membrane of the mitochondrion. We'll talk more about that. And we'll talk about mitochondrial anatomy at some point. I'm not going to talk about that right here. Well, actually, I guess I will. All right. See if I know what's on my lecture thing here. What you see on the screen is an electron micrograph of a mitochondrion. It's been sliced, and what you see is that the mitochondrion has a sort of a potato shape, at least in this particular figure. And it has, interestingly, a membrane surrounding it, and that membrane actually has two components. One, the outer membrane, and the other, the inner membrane. And of the two, the inner membrane is the far, far more important, as we will see later. We'll discuss that then. This membrane encloses a central chamber that we call the mitochondrial matrix. So the chamber is just that. It's just like a little opening. Okay. In this chamber is dissolved the equivalent, or is contained the equivalent of the cytoplasm, that is liquid contained in here. And it's in this liquid of the matrix that 7 of the 8 enzymes of the citric acid cycle are dissolved. So 7 of those 8 enzymes are dissolved in this liquid of the matrix. One of the enzymes is located in the inner mitochondrial membrane. All the reactions, of course, are occurring inside the mitochondria. That's where everything is occurring. One enzyme in the membrane, the rest of them dissolved in here. The other thing that you see here are some what are called infoldings of the mitochondrion. These infoldings have a name. They're called cristae, C-R-I-S-T-A-E, meaning they're plural. Okay? And these cristae have, as we will see later, within them a lot of complexes that help in the process of oxidative metabolism. So what these infoldings do is they're actually parts of the inner membrane. So they're infoldings of the inner membrane, and they're increasing the surface area of the inner membrane. When we look in cells that are going through a lot of oxidative metabolism, they have, number one, a lot of mitochondria, and number two, they have a lot of infoldings, meaning that they have a lot more surface area that's here. Yes, sir?
I was just wondering, the way it's pictured makes it look like little fingers. Is it more, because it's 3D actually, is it more fan-like?
It's a good point. It is 3D, and so it's not simply a two-dimensional infolding, but it is a folding, across 3D. So you can imagine this whole thing like a leaf almost sticking inside of there. Okay? Or like a hand, right? So it'd be more like this than like this, right? I'm not giving you the finger. Okay. That's bad, Professor. Bad professor. Don't do that. Okay.
So, I just want you to repeat that. Did cristae increase the surface area, which made it more efficient?
Cristae increase the surface area of the inner mitochondrial membrane, which basically provides for more complexes to actually be located. We'll talk about the complexes later. So the more surface area, the more we have, the more things can be there, present. And that'll become important, as you'll see later. Now, mitochondria, we know, are very important. For years, we've always said the mitochondria were the power plants of the cell, because it's in the mitochondria where most of the ATP of the cell is made. Okay? And we've known about that for almost as long as we've known about mitochondria. That's where most of the ATP is made. What we've come to realize in very recent years, meaning in the last 10 years especially, is that the mitochondria play very important roles in processes like apoptosis, cellular suicide, cellular aging, and a variety of things like that. And as we talk more about those, I hope to make clearer to you why these are very important for processes like that. Bottom line is that one of the things that these guys do is they use a lot of oxygen, and one of the byproducts of that use are reactive oxygen species. And we'll talk more about those as we get going along. Okay, there's a schematic of what I just showed you. Interpretive drawing, I think, as they say. The outer mitochondrial membrane does very little in the scheme of things. We will almost ignore it for our purposes. Now, we're going to start talking about metabolism. So if you recall, at the end of glycolysis, the product were two pyruvates for each glucose that we started with. 2 pyruvates for each glucose that we started with, okay? I told you that the entry point for the citric acid cycle was acetyl-CoA, and I said that it came from pyruvate. So now we need to talk about how we go from pyruvate to acetyl-CoA, and we have to see the enzymes and process that's involved in that. The oxidation of pyruvate is what produces acetyl-CoA. So even before we start the citric acid cycle, we have an oxidation, yet another oxidation. Pyruvate is getting oxidized to acetyl-CoA. That step, that very first step, is not part of the citric acid cycle. You'll notice it's up here in a box. You'll notice the citric acid cycle is down here. Don't confuse this first step with the citric acid cycle. It's required to get things into the citric acid cycle, but it's not a part of the cycle. This oxidation of pyruvate to acetyl-CoA occurs also in the mitochondrial inner membrane. Occurs in the mitochondrion, inside the mitochondrion. So that means pyruvate has got to be moved into the mitochondrion in order for this process to occur. Okay. How would you guys like to memorize that structure for the first exam? Okay. Now, you wouldn't, and I wouldn't make you memorize it, of course. What's that?
That was the whole exam.
How about extra credit? You like it? Is that what you said? You've got something on your foot, too. Did you kick somebody? Or is this for kicking somebody? These are pretty good for that. They're pretty heavy, you know. You not forgot that tan remark, have you? Okay. Acetyl-CoA is a molecule you're going to hear a lot about this term. I talked about it last term. Anybody remember the context in which I talked about it last term? What did I describe it as? It's an activated intermediate. It is an activated intermediate. And what was an activated intermediate?
Something that donated part of itself to something else.
It's something that donates a part of itself to something else using a high energy bond. This is the high energy bond. This is the acetate part, or the acetyl part. The rest of this thing is the CoA part, coenzyme A. Okay? This is a very high energy bond right here, and as we will see, this high energy bond helps this acetyl group to be donated to something else. Okay. Well, I promised that we would talk about the oxidation of the pyruvate. Let's get on to doing that. Pyruvate oxidation is catalyzed by an enzyme that has a little bit of a confusing name. It goes by two different names, and I will tell you the one that I prefer, and you'll see why in a bit. I prefer the name of the complex that does it, and the name of the complex is the Pyruvate Dehydrogenase complex. You can actually see the complex here. It contains many subunits, and I'll show you a schematic of this in a second. And these individual subunits catalyze different parts of the reaction or reactions that produce acetyl-CoA. It's a several-step process. Okay? So the name that I prefer for the overall process is the Pyruvate Dehydrogenase enzyme. Okay? Now the confusion is that one of the components of this overall complex is called the Pyruvate Decarboxylase. That's only a component of the overall complex. Your book, including some other books, occasionally slips and calls this complex the Pyruvate Decarboxylase complex. It's not. Okay? It's the Pyruvate Dehydrogenase complex, of which the Pyruvate Decarboxylase is a component. And I'll show you how that's the case as I show you the mechanism of the reaction in a minute. Okay? Are we clear on that? Okay. Here are the players in the complex. Okay? The players include, and then see your book has already slipped right here, they call it the Pyruvate Dehydrogenase component. It's actually the Pyruvate Decarboxylase component. And by the way, I'm not going to ask you to memorize these. If you know them as E1, E2, and E3, I will be more than happy. Okay? So there's three components in that multi-subunit. There's several pieces of each one, several copies of each one in that overall complex that I showed you. 24 E1s, there's 24 E2s, and there's 12 E3s. That's a lot. That cube that you saw there in that electron micrograph is fairly large. It has a lot of proteins in it. Each one contains a separate coenzyme. Anybody remember what a coenzyme is from last term? So it's a non-amino acid that associates with the protein and helps enhance that enzyme's function, or that protein's function. It doesn't have to be an enzyme. Hemoglobin had a sort of a coenzyme of heme, for example. Okay? Well, these guys here are 3 coenzymes, one associated with each, the E1, the E2, and E3. Okay? Each one of these, as we shall see, is part of a 3-step reaction that produces acetyl-CoA. The first one's involved in the oxidative decarboxylation. And that's, again, not completely correct. If you notice the decarboxylation, you're in better shape. The actual oxidation occurs at this step, the second step, not the first step. And FAD is involved in carrying the electrons. And again, I know it's a little vague. I'll show you the steps in a second. Okay. Here are 2 of those coenzymes. The first one's called thiamine pyrophosphate. Yes, it's related to the vitamin. We get it from the vitamin thiamine. Okay? And thiamine pyrophosphate plays a very important role. The main role that this thiamine pyrophosphate plays is it carries a 2-carbon piece. It carries a 2-carbon piece. That's basically what it does. All right? We'll see that it carries it and it transfers it over to lipoic acid. So first it grabs it. During that grabbing process, there's a decarboxylation that occurs. It passes it over to lipoic acid. And then lipoic acid releases it and passes on electrons. Okay, let's see how this goes. All right, so this now shows us the 3 steps in going from pyruvate to acetyl-CoA. Pyruvate has 3 carbons, acetyl-CoA, the acetyl part, has 2 carbons. Okay? The first step in the process involves decarboxylation, meaning we're releasing a CO2. Well, if we start with 3 carbons and we release 1 carbon, we have 2 carbons left. And rather than release that 2-carbon piece, our cells grab a hold of it with thiamine pyrophosphate and they pass it over to the next place where the reaction will occur. In other words, it's going to pass it over to the lipoic acid. So we think of thiamine pyrophosphate, we think of lipoic acid, and over here we think of, well actually the electrons here go to electron carriers. That's where those electrons are going. Okay? Once the electrons have been carried by something else, acetyl-CoA is released. Now, the overall enzyme complex, I've shown you 3 coenzymes. It turns out there's a total of 5. TPP, okay, lipoic acid, coenzyme A, there's 3 of them, NAD and FAD, because they have to carry the electrons. So we have 5 coenzymes associated with this enzyme. Now I'm going into some detail here because it turns out that the mechanism that we see in this reaction is duplicated in another reaction of the citric acid cycle. Okay? So what you see going on here is duplicated in another reaction of the citric acid cycle. Well, why am I so picky about pyruvate decarboxylase versus pyruvate dehydrogenase? What's the big deal? Why do you care? Well, I'll tell you why I care. You guys all care about this. If you drink beer, you care about this. Okay? Because we talked last term about how it... Come on in. We talked last term about how it was that ethanol was produced, right? They'll tell you later what the joke is, okay? We didn't do that to embarrass you. That's bad to do that on first class. Bad class, okay? Bad to do that on the first day. I'll spank every one of you, okay? No, we're not going to do that either. I really would be bad professor then, wouldn't I? Oh, wasn't that break nice? All right. So why do we care about the decarboxylation? Well, it turns out that this process that you see here is what occurs in our cells all the time. In bacteria and yeast, they are capable of decarboxylating without going through the remaining steps. So what they produce as a result of the decarboxylation is a molecule called acetaldehyde. It's a 2-carbon molecule, and the reduction of acetaldehyde by NADH gives you ethanol. Anybody want to remember why acetaldehyde had to be reduced? What was important? Why was that reduction important?
Regenerating NAD.
Regenerating NAD. Okay? This happens when the bacteria don't have oxygen. They have to regenerate their NAD, and they do that by donating electrons to acetaldehyde to make ethanol. So in bacteria and yeast, when they don't have oxygen, they will stop after the decarboxylation. That means, therefore, that this decarboxylation is not an oxidative decarboxylation. The oxidation occurs later. So it's one of the few decarboxylations we see in cells where the decarboxylation itself is independent of the oxidation. Or it can be in bacteria and yeast. In us, they're linked. We have no choice. In us, once we decarboxylate, we always go on through and finish that process. Okay. Here is that ring group of thiamine pyrophosphate. We're not going to go into mechanism here. Don't worry about this. All right. I'm just showing you what's here. All right. What's happening is here's the 3-carbon molecule pyruvate. Here is a carboxyl group that's getting ready to go bye-bye. It goes bye-bye, and this 2-carbon piece becomes attached to thiamine pyrophosphate. That's important because, as I said, thiamine pyrophosphate is the carrier. It's grabbing this 2-carbon piece, it's not letting it go anywhere, and it's going to transfer it over to the lipoic acid. Okay? So the decarboxylation happens, this gets attached and transferred. This guy here actually has a name, and I think your book actually calls it the name I use it as well, and that is, it calls it an activated acetaldehyde. It's like an acetaldehyde, but it's an activated acetaldehyde. So this 2-carbon piece is going to be passed over to lipoic acid.
Why is there carbanion there?
I'm sorry?
Where did that carbanion come from? It's not just floating around. So.
Where did the...
Coming from thiamine pyrophosphate, when did it become a carbanion?
Oh, how did this... Okay, so a very good point. This guy right here, right? How did that happen? Okay, so again, we saw in serine proteases, we saw activation, we saw abstraction of protons. The same thing has happened here to get this. It's a good question. Has happened to get us to this point, but I'm not showing you. Again, I'm not showing you the mechanism for that. But good, very good eyes. Good question. Okay, let's see. There's a mess. Nah. Okay. So, as I told you, that activated acetaldehyde gets transferred over to lipoic acid. And what you see happening here is, actually, I do need to show you one thing off that last figure. Sorry about that. It's actually this one. Okay. So this shows us the transfer of the 2-carbon piece to the lipoic acid. You notice it's called lipoamide. I use these terms interchangeably. Okay. It simply means that lipoic acid has been attached to a lysine in the enzyme. That's when you get the amide part. But the functional part of the lipoic acid is this guy right here. It's a disulfide bond. You saw disulfide bonds, and we talked about protein structure last term. And when I had a disulfide bond, is it in the oxidized or the reduced form? It's in the oxidized form. Okay? In order for it to be a sulfhydryl, it has to be reduced. For something to be reduced, what has to happen to it? It has to gain electrons, right? Well, where are the electrons coming from? They're coming from this activated acetaldehyde. It's transferring its electrons to this bond. That's how we're generating this sulfhydryl over here. We've broken that ring. That requires electrons. The electrons are coming from this reactive acetaldehyde, which is becoming oxidized. You see it's an alcohol here. You see it's a double bonded oxygen over here. This is where the oxidation is actually occurring in this process. The disulfide is becoming a sulfhydryl. So here's the oxidation that's occurring in the process. This guy right here is now electron rich. Well, if we're going to keep catalyzing reactions, we've got to get this guy here back to this form. Which means we've got to get rid of the electrons that the lipoamide has gained in this process. This is what happens next. Okay? Here's this electron-rich lipoamide. It has the acetaldehyde attached to it. Okay? It's actually an acetyl group at this point. Notice that the acetyl group is donated to a CoA. So this is just simply a transfer over to the CoA. This creates acetyl-CoA. And lipoamide, which is fully reduced. It has SHs at both places. We've got to get rid of those electrons. Right? So we passed off the acetyl group to a CoA, which now can go out and go to the citric acid cycle. Next, this guy right here takes its electrons and donates them first to FAD. It donates them first to FAD. That creates FADH2. It also recreates that disulfide bond. So the enzyme can now go back and recatalyze another reaction. But this FADH2 does something odd that we don't see happen in cells. It's donating electrons to NAD to become NADH. This is how this reaction actually produces NADH. It's kind of complicated. It's rare that we ever see this process occur in cells, and I won't say more about it unless you have if you have questions about that, see me later and I'll tell you more about that. But suffice it to say that the electrons at the end of this overall process are residing in NADH. Well, what did we say about NADH last term? What about NADH and NAD? They're interchangeable, right? And so if I have a lot of NADH, what happens to NAD? Concentration goes down. Concentration goes down. What's going to happen to this reaction? Probably not going to occur, right? Okay, so like we saw last term, we've got to keep the NAD concentrations high in order to keep this process going. The difference is, last term, that conversion of pyruvate to lactic acid that kept our NAD going, where did that occur? In the cytoplasm. This is occurring in the mitochondrion. They're not linked, okay? So if we run out of NAD in the mitochondrion, we stop this process. We'll stop citric acid cycle as well, okay? And there's no way around it. Okay, I thought I saw a hand back in the back. Did you have a question back here? You forgot it. I talk too much, don't I? Okay. Okay. So that is this process. There's lipoamide, and again, it's called lipoamide because it's linked to a lysine side chain of the enzyme itself. That's the E2 component of the enzyme. Here's a schematic of that, what you saw in the figure earlier, and no, you're not going to draw that. Unless you want to, of course. And there's the reaction, blah, blah, blah. We actually start the process here. Pyruvate gets decarboxylated. We see the thing transferred to thiamine pyrophosphate, which now passes it off to the lipoamide, which happens right down here. Okay? The lipoamide passes it off to acetyl-CoA, to a CoA group to make acetyl-CoA. and then the electrons just get passed to FAD, and then to NAD, and we're back where we started. Okay? That's all there is to that. Okay, questions about that before I move on to the citric acid cycle? Nobody? Yeah.
Yeah, so for the carbanion plus pyruvate to hydroxyethyl TPP, what enzyme is that? E1?
So that is E1. E1, yeah.
And then the hydroxyl TPP plus lipoamide to the
Right. So wherever you see TPP involved as a carrier, that's E1. Where you see lipoamide involved as a carrier, that's E2. Okay. And when you see the rest of the stuff, that's all E3. Okay. Okay. Well, let's turn our attention to the citric acid cycle. And believe me, this is not going to be a term of mechanism. I know it's a bad mechanism to start with, but that's where we are. We're not going to spend a lot of time talking about mechanism, so you can relax if you don't like mechanism. Okay, the citric acid cycle is, as I said, the major oxidative pathway of cells. It doesn't have a beginning or it doesn't have an end, but traditionally we talk about the beginning as the entry point for the acetyl-Coa, because that's the sort of gasoline that keeps the overall cycle running. Without acetyl-CoA, we can't run the full cycle. Okay? Well, here's oxaloacetate, which is floating around inside the mitochondrion thanks to a variety of processes, one of which includes gluconeogenesis, one of which others include amino acid metabolism. But suffice it to say, we've got plenty of oxaloacetate floating around inside of the cell. Okay? We've just produced acetyl-CoA by the decarboxylation of pyruvate, by the Pyruvate Dehydrogenase complex. And in this reaction, which is catalyzed by an enzyme called Citrate Synthase, and yes, you need to know the enzymes for this, Citrate Synthase catalyzes the joining of the acetyl group, and by the way, forget this intermediate, we're going to just forget that exists, all right, to make a citrate. There's the 2 carbons from the acetyl, all right, there's the 4 carbons from the oxaloacetate, and we make a citrate.
What was the enzyme called?
So I'll give you an example. That's what I'll finish with today. It turns out that this enzyme right here, a conatase, is strongly inhibited by Fluoro Citrate. Fluoro Citrate. Well, what's Fluoro Citrate? Is that something I get when I brush my teeth? No, it's not involved in brushing your teeth. The fluoride in your teeth doesn't give you Fluoro Citrate. It turns out that back in the 60s, there was a sort of a miracle poison that people came up with to kill coyotes out in the western range. And it was a compound called fluoroacetate. Fluoroacetate is an acetate that has a fluorine in place of one of the hydrogens. And it was very, very good at killing coyotes. Well, why was it good at killing coyotes? Well, that fluoroacetate could readily be made into fluoroacetyl-CoA. That enzyme didn't mind it. The fluoroacetyl-CoA could donate the fluoroacetyl group to an oxaloacetate. The citrate synthase enzyme didn't mind it. However, the aconitase enzyme, when it got fluorocitrate, went and it stopped. That effectively stopped the citric acid cycle. Well, you stop the citric acid cycle in a cell for very long, you kill the cell, you kill the organism, because if all the cells are dying, that's what happens. So this is how it killed coyotes. It turned out, however, that there were that something that was designed to wipe out coyotes unfortunately made it into the food chain, because vultures and other organisms that were out there that would pick up and eat coyotes and things that ate them started passing fluorocitrate through the ecosystem and causing major problems among bird populations. Major problems. As a consequence of that, fluorocitrate's use was significantly restricted for many years, and I believe is finally banned. It cannot be used and now, because it does persist and gets picked up by organisms that eat the dead organisms that are around. Okay, just a little sidelight for you. We're finishing 2 minutes early. It's a good omen for the term. So, see you guys on Wednesday. Set it up. Just come see me sometime this week and we'll get it set up. Okay?
Cool. Awesome. Thank you. Thanks.
Hey.
Hi.
So are your office hours still kind of whenever you can fit people in?
Just check my schedule and come on in.
All right.
Okay? Take care. Hi, this is Kevin in Gilthorne Auditorium. The wireless microphone in here was not working during my lecture. I swapped the batteries and it still was dead. So it would work for about 2 seconds and then it just stopped. Yeah. It did start, but it would last maybe a minute and then the whole thing was gone, even when I swapped the batteries. You might want to get some new batteries or a new microphone, one of these. Thank you. Bye. Hi, how are you?
Good, how are you?
Good.
I took the 450 last year.
Yes?
So I'm wondering, I just want to know what you recommend to review.
To review? Look over the material. You took it last year. The material's still online. You can look. I would look through that and just get an idea on that.